Hey students, this time we're going to be talking about how are genetic diseases inherited. We're going to be talking about the idea of inheritance. And when we get into that, we need to know that genes are the basic unit of inheritance. We mentioned previously that genes are parts of our DNA. We have just under 20,000 different genes in our genome. The genome is, what we call all of our genetic information. Recall that we have 46 total chromosomes, and of those 46 chromosomes, we have, 23 pairs of chromosomes. And on these chromosomes, we have different types of genes. So let's say that we have, here we have a chromosome that we got from our mom, and let's say that here we have a chromosome we got from our dad. Now on this chromosome somewhere, maybe there's a gene here for eye color, and from our mom we get green eye color, but from our dad we get blue eye color. Now, the similarities between these two things is that they're both eye color, but we have one green and one blue, and that would be an allele. It's a variation of the gene. And these variations are usually due to small changes in the nucleotide sequence, like we mentioned before, whereas, here we get green and blue. And examples of eye color are, alleles are for eye color, blood type. Some people have, A blood, B blood, AB blood, and there's all kinds of different ways that you can get alleles. Well, what is a chromosome really? We mentioned a chromosome. Chromosomes are these long pieces of DNA wrapped around proteins called histones. We call this DNA wrapped around a nucleosome, and then those are compressed together to form a chromosome. The chromosome is compact, but it can be unwound to provide access to individual genes. And so I'm going to draw, typically DNA is, of course, double-stranded. We know that. And you have the nucleotides between. I'm going to reduce this down just to a little tiny thread here. So typically our chromosomes, they can be unwound and very thin, but in order for this chromosome to fit, and remember we have 46 of these, in order for that to fit in our nucleus, we have to take this chromosome and we wrap it around these little things called histones. And so what you end up with is something that looks a lot like this, like thread wrapped around a spool. And then we take this and we wrap it even more around itself. And so really, your DNA is pretty packaged inside of your nucleus sometimes. As a matter of fact, if you took all 46 of your chromosomes and you laid them end to end, they would be over 6 feet long, which is a lot of chromosomes in there. So here we see, a much better drawing than what I did, but we see our nucleosomes in here. We see the nucleosome there. We see our little histones there, and all of this kind of wrapped together into a chromosome. Now this is showing a chromosome that's been condensed and duplicated, and so what we have are sister chromatids. Typically inside of your cell, and so here we have our cell, we have our nucleus there, you'll have a chromosome from your mom, let's say, and then you'll have a chromosome from your dad. And before we go to copy this, we need, before this cell will divide into two new cells, we have to make a copy of each of those chromosomes. And so you end up with 2 from your mom and 2 from your dad. And that's where these are called sister chromatids. Humans and a lot of other organisms actually are what we call diploid. Di meaning two, employed refers to the chromosomes. And so we have two copies of each chromosome, one from our mom, one from our dad. Diploid is often abbreviated 2N, with N referring to the number of chromosomes. In the individual, humans have 23 of these, and so 2N means we have two copies of each for a total of 46. The various pairs of similar chromosomes are called homologous chromosomes. So if we look at, the chromosome from your mom, the chromosome from your dad, they're very similar to one another in that, here we have an eye color gene of some type, and here we have an eye color gene of some type. So that makes them very similar. But remember, I think we got green eyes from mom, and we got blue eyes from dad, maybe. I can't remember which one we had. But so they're similar in that they both have eye color genes there, but they're different in that they have different alleles. We can see this here. So this, these two are homologous chromosomes, and these, I got to figure out how to just point. So this is homologous chromosomes here, but these two are sister chromatids. How are sex chromosomes different? All right, well, this is a human karyotype here, and so what we see is that we have 22 types of chromosomes, numbering 1 to 22, and then we have these kind of special chromosomes down here called the sex chromosomes, and a female is XX, and a male is XY. So be able to make sure you can go through that. What is sexual reproduction, and specialized gametes, and so we'll see that. Humans have two copies of each chromosome. We're diploid, and so we know what diploid means. We have two of every type of chromosome, and we see that here. So we have two of every type. One we got from mom, and one we got from dad. During sexual reproduction, the organs of the reproductive system produce gametes. Gametes is an important word. Eggs are gametes, so are sperm cells. Eggs and sperm are what we call haploid, or single N. And the process that does this is called meiosis. So let's start with here. So we have two diploid adults. So this is diploid, and this is diploid here. And both of these adults are diploid, which means that inside of their cells they have two complete sets of chromosomes. So they have one from mom, one from dad. But during meiosis, right? So when meiosis happens, each one of these individuals will then produce a cell. Will produce a cell that only has either the one from mom, or it will produce a cell that has the chromosome from dad, and that means it's haploid. So the haploid egg will then fertilize the haploid sperm. You end up with a zygote and a diploid baby. So meiosis is a form of cell division that reduces the chromosome number of the daughter cells from haploid to diploid. And in humans, what that means is you have a cell with 46 chromosomes. That cell will go through something called the S phase, where it will replicate its chromosomes, and you'll end up with 92. This cell that has 92 will go through one stage of meiosis, and it will produce one cell, two cells with 46. This cell will then divide again in meiosis part 2, and you'll end up with 4 cells that have 23 apiece. Well, meiosis follows the same series of steps as mitosis. So you have prophase. So you have prophase here, metaphase, anaphase, and telophase. But notice that it's meiosis 1. And meiosis 1 is followed by meiosis 2, where we have prophase, metaphase, anaphase, and telophase again. Well, in humans, meiosis only occurs in the testes or the ovaries. In males, this process is called spermatogenesis, and oogenesis is the term used to describe meiosis in females. Oogenesis is slightly different than spermatogenesis in that the output will be a single haploid egg, whereas in spermatogenesis, you end up with 4 haploid sperm. What are the general patterns of inheritance? So Mendel did some important work. He was an Austrian monk who performed some classic experiments on pea plants. He knew nothing about genes, and he knew nothing about DNA. But he had a background in in science and math, and so he worked out how things can be inherited. In this crossing, what happened was he took a homozygous plant. All right, so what does homozygous mean? Well, homozygous means that this green potted pea plant here, if we looked at its chromosomes, so we looked inside. All right, I'm going to stop drawing the cell. I hope you guys don't mind. Just know that chromosomes exist inside of a cell. So this plant has 2 of every type of chromosome, and somewhere on this chromosome is a gene that determines what color the seed pod is. And in this case, it's green and green. Both of them are the same. Since this thing has both of these the same, then we call this homozygous. This one here has 2 of the same as well, but they both code for yellow. Now, this parent here can only pass on one of these chromosomes. It can pass on this one or this one. Well, in this case, it has to pass on a big G. Same thing for this parent. It has to pass on a little g. So therefore, what we make in this first generation is a heterozygous plant. But when this plant is crossed with itself, so what Mendel did was he allowed big G, little g plants to be crossed with big G, little g plants. So now this parent can pass on a big G sometimes here, or it can pass on a little g sometimes here. This parent can pass on a big G, or this parent can pass on a little g. And now what you do in this Punnett square is big G, right? And this big G goes here, this little g there, and if we look at it again, big G goes here, so there it is, and little g, there it is, and you can figure out this one because the little g and little g go there. I couldn't make the slide go. Well, Mendel's pea plants were important. He studied lots of different kind of traits, and he looked at stem length, tall and short. He looked at inflated, constricted, round, and wrinkled, axial, terminal. This is kind of a classic one here, purple and white, but he studied all kinds of different patterns. Mendel, through his work, developed this thing called the law of segregation, which states that each individual, so me and you, has two factors for each trait, which means that we have two of every chromosome. And these factors separate during the formation of gametes. So he knew that we had two somethings, and that during the formation of gametes, those factors were cut in half. Not cut in half, but each gamete got one or the other. Well, the principles of inheritance gives us some way to look at pedigrees too. And in pedigrees, males are squares, so this would be a male. Females are circles. Anything that's shaded represents individuals expressing the genetic trait. And then you have lines kind of connecting people. So here what we have is an affected female married an unaffected male. Well, if we look at that, we can see that these parents had two offspring, both girls, and both of these girls, this one and this one, were unaffected. And they also married males that were unaffected too, and they had a female that was unaffected, and this female married this male. All right, so this is kind of weird because this is their first cousins, it seems like, and these two, this female and this male, then had two children that were affected by this genetic disorder. Here we see another pedigree kind of filled out, and you can see how this worked. And in this case, heterozygotes are affected. So if you're little a, unaffected. But if you're big A, you're unaffected. But if you're big A, little a, it means that you have the disorder. Are all traits recessive or dominant? Well, the phenotype is the result of the proteins produced by the gene, right? So when we talk about the genetics of something, let's talk about Mendel's pea plants. And so Mendel, you could either have purple flowers or white flowers. And somewhere on that pea plant, you had chromosomes. And if you had big P, you were purple. If you had a big P, you were also purple. And if you had little p, your flower colors were white. Well, what happens in Mendel's pea plants is that this gene here produces a protein that's purple in color, right? And so this kind of spits out purple protein. This one spits out purple protein. So it makes sense that the flower would be purple. So we understand that one. If you're big P, little p, if you're big P, little p, this one spits out purple protein. But this little p is broken. It doesn't do anything. So therefore, the color you end up with is purple. Well, if this one is broken, it does nothing. This one is broken, does nothing. So therefore it gets no color and no color. So the flower color is white because this is broken. Well, in this case, that is what we would call complete dominance. But there's also this example of incomplete dominance where the dominant allele does not completely mask the recessive allele. And so you end up with things where if you're, well, let me illustrate it without that example real quick. There's a flower that, another flower where if you have red and red, then you have a red flower because this makes red and this one makes red. If you have white and white, you have a white flower. But if you have one red and one white, it makes pink. Because this makes red and this one makes white. And these are in snapdragons, by the way, not pea plants. So in this case, this white allele makes the white color and they mix up. Hypercholesterolemia, individuals who are heterozygous have an intermediate phenotype, a characteristic of incomplete dominance. So plasma cholesterol. So if you are normal, then you have a normal range. If you're heterozygous, you have these cholesterol deposits. And if you're homozygous, then you have a lot of cholesterol deposits. So that would be incomplete dominance too. So what if there are multiple alleles for a gene? So, when we talked about Mendel's pea plants, we were talking about, big P or little p, red, white. But what if, what about blood types? So in blood types, you can have A, B, or O. And so now that's multiple alleles. And you still only have two of each type. So if you're type A blood, then you have one chromosome with A on it and one with O, or you can have one chromosome with A and the other chromosome with A. And that means that you have A blood too. Sometimes the environment plays a role too. So it can affect the expression of genes associated with like skin color shown here. Skin color is determined by 3 genes, A, B, and C. And if you have the recessive version of all 3 of those, then you have 0 dominant alleles and your skin color is pretty light. If you have all the dominant alleles, then your skin color is pretty dark. Genes can interact to produce a phenotype too. So in some cases, one gene affects the expression of another one. So how does this relate to sickle cell disease? Well, here we have a female carrying the sickle cell gene. Here we have a male carrying the sickle cell gene. The mom passed on her sickle cell gene, and the dad did too, to this individual. And in this case, in the case of the second child, this child here got this and this. This one, the next child, could have gotten his from here and this from here, so they're just a carrier, so forth and so on. And you can look at the rest of this pedigree and kind of figure out where the sickle cell disease comes from. And that is it for this chapter.